Okay, hello everybody. Welcome to the final exam review. So without any further ado, let's get right into it then. So this is going to be a fully cumulative exam where you're going to be expecting to get questions from the entire course. And so we need to go all the way back to the chapter 11 content on the nervous system. And one of the first things you talked about was the basic neural pathway. And it composed of 5 parts. Begins with the sensory receptors that receive the stimulus. A stimulus is a physical response, and the sensory receptors are a modified neuron that converts that sensation into an action potential. The second part is the neuron that will take that action potential to the central nervous system. So the sensory neurons transmit impulses from the sensory receptors toward the central nervous system. The interneurons are the neurons of the central nervous system, the spinal cord, and the brain. And the motor neurons are the neurons that conduct impulses from the central nervous system out to the effectors. The effectors are either going to be muscle cells or glands. Ultimately, they are the response that the central nervous system sends, and the effectors is the last part of that basic neural pathway. So it's important to remember the order they go in and try to visualize that order as well. This is a picture version of the same thing where we have the sensory receptors. In this case, they are touch receptors of the skin. They convert that physical stimulus into a nerve impulse. The physical stimulus is converted into that action potential that travels along the sensory neuron. Now, you don't have to differentiate between what a sensory neuron looks like versus what a motor neuron looks like, but just be aware that the definition, as far as we're concerned, of a sensory neuron is one that conducts a nerve impulse toward the central nervous system, as opposed to a motor neuron that conducts a nerve impulse away from the central nervous system. The interneurons make up the neurons of the central nervous system, and in this basic diagram, we just see one interneuron, but there can be multiple interneurons. So in this general basic neural pathway, we have 5 parts, but really the interneurons, we can see them connecting either directly between the sensory neuron and the motor neuron, or you can see multiple interneurons as they send or conduct the impulse sensory information to the brain. This is an important thing to try to commit to memory as well. It's the processes or the steps that are involved in continuing or propagating the action potential from the presynaptic membrane to the postsynaptic membrane. So we're going in this direction. Well, I would be if I could get this thing to draw. There we go. So we're going in this direction. And at the synaptic terminal or the synaptic cleft, that 20 nanometer space between the axon terminals of the presynapse to the dendrites of the postsynapse, we see the release of neurotransmitters. So the neurotransmitters are stored in the vesicles. The neurotransmitters engage in what they call exocytosis with the presynaptic membrane, and it spills the neurotransmitter contents into that synaptic cleft. Now, those neurotransmitters can either be taken up by the postsynapse receptors, and these things are in constant states of motion, so they can attach to them, but they can be shaken away as well. The more neurotransmitters, the more likely we're going to see attachment to those receptors. And to make sure that we don't have too many neurotransmitters, so that those receptor sites do have an opportunity to become free of neurotransmitters, we're assisted by these degradation enzymes that break up the neurotransmitters. Or very simply, these neurotransmitters could just simply diffuse out of the synaptic cleft. Or there's reuptake as well, where they could be taken up by the presynapse and reused. So it's important that these neurotransmitters are not kept in the synaptic cleft for too long. Because let's say, for example, the cellular response here, let's say it's to cause localized depolarization. In other words, as soon as that neurotransmitter attaches to the receptor site, then it causes the sodium ion gates nearby to open up. Sodium ions rush in, causing depolarization. Now, in order to allow for repolarization to occur, we need to close those sodium ion gates, which means we need to liberate this receptor of those neurotransmitters. So keeping the neurotransmitters only briefly in the synapse is very important for the recovery of the post-synapse response. So hopefully this is getting very familiar to you guys. And of course, I strongly recommend you watch the other review videos as well. You know, there's a lot of review stuff that you have access to here. So in this curve, we say time in milliseconds along the bottom, and then we have a millivolt response along the side. Negative 70 millivolts is the resting potential of this particular neuron. And you should never get married to these numbers. I mean, these numbers really relate to the neuron of a giant squid. You know, they're going to vary from one neuron to the next. So don't assume that all resting potentials have to be negative 70 millivolts. They just happen to be on this diagram. As the sodium ions begin to rush in from an adjacent part of the neuron, we start seeing an increase in potential. And when it hits about negative 55 millivolts, that's enough sodium ions to bring up the potential to open up the sodium ion gates, allowing in generalized depolarization. So those sodium ion gates are voltage-mediated gates, meaning they open and close depending on what the voltage is. So at negative 55 volts, they open, and those sodium ions rush in by diffusion and charge attraction. Then according to this diagram, it's about what plus 35 millivolts when the sodium ion gates close. And at the same time, the potassium ion gates open. Now the potassium ions are on the inside of the axon. And so they're going to be flowing out, causing repolarization. Now those potassium ion gates don't close until we get much lower than the negative 70 millivolts. In this diagram, it looks like it's about negative 80 millivolts. So this hyperpolarization is important because it means it requires an awful lot more sodium ions to breach the threshold potential. So this prevents a premature response at that part of the membrane. And we don't want to have a premature response because the ions are in the wrong places. The sodium ions are on the inside, the potassium on the outside. So we need some recovery or refractory time in order to reestablish the ions on the right side of the membranes so we can handle another action potential. So hyperpolarization is, and then this was one of the written response questions from the first unit. Why is hyperpolarization important? There's two reasons. Basically, it's important because it needs to engage a refractory period, a period of time where we're prevented from a premature action potential because of the large distance it is between hyperpolarization and threshold levels. The other reason, of course, is it makes sure that the action potential goes in one direction, because along an axon, you can see that, if this is an axon here, well, I could draw it, but this stupid thing isn't letting me. You know what? Forget the axon. I'm not going to draw it. Just take my word for it. It prevents it from going in the other direction. All right. Also part of chapter 11 is a discussion on brain anatomy. And it's very important to know what parts of the brain are. The largest part of the brain is the cerebrum. And the cerebrum is divided up into lobes. But if you're asked some general things about, consciousness and thought and behavior, memory, that kind of thing, we're talking about the cerebrum in general. The lobes, they're important to remember what the lobes are. The frontal lobe is the primary motor area. So that's the important bit, the primary motor area. The parietal lobe is the primary sensory area. The visual area is in the occipital lobe, and auditory association is in the temporal lobe. Now, all the other bits and pieces in this diagram from your book, you don't need to memorize. It's just important that when it comes to the cerebral lobes, just know what the general part is. Visual information goes to the occipital, auditory information goes to the temporal. Sensory is distributed to the parietal, and motor to the frontal lobe. And this diagram also from the book, again, there's a lot more on here than you need to memorize. Something that probably doesn't get as much discussion really is the brainstem, which is made up of the pons, the medulla oblongata, and the cerebrum. And they all have very important parts, particularly the medulla oblongata. That has some autonomic control as well. So such as, for example, breathing rate and heart rate, blood pressure, that kind of thing is moderated by the medulla oblongata as part of the autonomic nervous system. Remember, the other part of the autonomic nervous system is the hypothalamus. So there's other autonomic functions that are associated there as well. Notice as well in the brainstem, it really is a continuation of the spinal cord. Any injuries of the spinal cord, while can be paralyzing to the body, actually generally doesn't seem to affect the face. Individuals, for example, who have spinal injuries will still have, an ability to be able to move perhaps their head and those facial features, still engage in conversation, speech, and so forth. Generally, any impacts to individuals with regards to ability to move muscles in their face or speak, it's usually brain injuries. And speaking of the autonomic nervous system, make sure that you're comfortable being able to tell the difference between the parasympathetic and the sympathetic branches of the autonomic nervous system. The parasympathetic, the so-called rest and digest response, works in opposition to the sympathetic nervous system, the so-called fight or flight. So if your body needs to be engaged in an activity to, in a preparatory response for an emergent situation, you're going to get the increase in heart rate, breathing rate, increase in blood pressure, all the associated physiological responses to deal with that emergency. That's part of the sympathetic nervous system. But once the emergency has gone away, it's not enough to actually just take away or reduce the sympathetic nervous system, but we also need to increase the parasympathetic nervous system. Now, I've always found that you don't really need to memorize the actual physiological responses to these two systems, because all you have to do is just think about it. You do need to know what they do, but if you're part of the sympathetic response, What is your body doing in order to deal with an emergency? Is your heart rate increasing or is it decreasing? Is your breathing rate increasing or decreasing? These normal responses, we can probably um, answer these types of questions without even having to worry about memorizing the specifics. Okay, chapter 12 is the sensory reception chapter. You focused on the eye and the ear, and your final exam is really going to focus on the eye and the ear. Basically, this diagram from your textbook, if you know what this diagram, if you're able to label this diagram, then you're set. And it's also not enough to label, but know what the structures do, the general structures. We're not going to give you a question in the exam. What specifically does the ciliary muscle do? Well, I don't know. I mean, we're looking for something that's going to be a bit more specific to the vision process. For example, you would have to be very comfortable with the notion that the retina is the inner layer of the eyeball that contains the photoreceptors, the rods and the cones. The cornea, for example, is a transparent part of the sclera. It acts as a fixed lens, bending light toward the center of the eye. And the sclera itself, a very thick, protective outer layer. If you had an opportunity to have a look online for the eyeball dissections, hopefully you'll see a video that will show just how tough that outer layer is. And the pupil, of course, is the space that the iris makes when it dilates or constricts to limit the amount of light coming into the eye. And the humors, we've got an aqueous humor at the front of the eye, which is a watery solution that is replaced. And we got a thick gelatinous humor toward the back there, the vitreous humor. And of course, the optic nerve, where is that going to terminate? It'll terminate in the occipital lobe of the brain. And the ear, you would have spent some time on the ear. Again, the structures of the ear is extremely important. You don't have to know the individual names of the ossicles. Just the ossicles is fine. Also, the inner ear, which contains structures that are fluid-filled, such as the cochlea, that contains the hearing hardware. This is where the actual vibrational motions are converted into action potentials and sent to the temporal lobe of the brain by way of the auditory nerve. And the semicircular canals as well. The semicircular canals in the vestibule are responsible for interpreting body position. The vestibule is for static equilibrium, in other words, head position, whereas the semicircular canals are the ones that are going to be responsible for communicating to your brain body position. You have those three semicircular canals arranged in different planes of space, so they're able to communicate to your brain your body position. If there's any disruption with the semicircular canals, it's going to cause dizziness and disorientation. Sometimes we look for that when we go on amusement rides at parks, but also it can be problematic for individuals who suffer from things like vertigo. Chapter 13 goes into hormones. There's a lot of hormones that you would have had questions on in the midterm exam, and of course the unit exam that covers this. So you shouldn't expect too many questions in the final on this stuff, but there are going to be a few, and your comfort with this information is important. This picture from your book, I think it's from your book anyway, is for ADH. the antidiuretic hormone. This is produced by the hypothalamus, but released by the posterior pituitary gland. And this job ultimately is to target the kidneys to reduce urine output. So ADH then increases water reabsorption. And as soon as the cells of the hypothalamus indicate that the body is becoming more hydrated, it will send a feedback message to prevent the release, a further release of ADH. The anterior pituitary gland secretes human growth hormone. And the growth hormone is responsible for increasing muscle mass and bone density. It's extremely important as well. It's released it continuously throughout life, but at certain times during childhood, during adolescence, and in combination with sex hormones, it's released in higher amounts, and that's responsible for things like growth spurts. This was a feedback pathway for thyroid hormones. For the questions that I marked for the individuals doing the Chapter 13 unit exam on hormones, I did find, and also the Chapter 14 questions on hormones as well. When asked about the feedback pathway, there was a number of you that really had problems with being able to articulate what this feedback pathway is. So maybe practice with being able to say out loud, what a feedback pathway involves. Basically, in this case, the hypothalamus is releasing a releasing hormone, thyrotropin releasing hormone, and targets the anterior pituitary gland, which responds by secreting thyroid stimulating hormone, or TSH. So that's a tropic hormone that targets the thyroid gland, which responds by releasing thyroid hormones. Thyroxine is the most common hormone, or T4. And most cells in the body will respond to thyroxine. Now, there's a negative feedback pathway attached to thyroxine and TSH. When enough thyroid hormones have been released into the circulatory system, they start connecting to the receptor sites on the anterior pituitary and the hypothalamus, and that triggers those structures to turn off their hormones. So the hypothalamus will turn off TRH, anterior pituitary will turn off thyroid stimulating hormone. And also in this particular diagram, it shows that TSH is part of a feedback mechanism as well. When TSH releases enough hormone into the circulatory system, then it attaches to receptors on the hypothalamus to switch off TRH. So what happens if our body produces lots of TSH? If TSH levels go up, then so do the thyroid hormones. What if there's something wrong with the thyroid gland? For example, it's no longer able to produce thyroid hormones. What happens to TSH then? Well, in this particular diagram, the TSH levels are actually moderated by its own negative feedback. But what if it's not? If TSH levels are allowed to be released, and if the only thing that switches off TSH levels is this negative feedback, oh, for goodness sake, it's not letting me draw. Maybe I'm drawing too quickly. There you go. If I just slow down. So if there's no thyroid hormones, then TSH levels are going to continually grow. Remember my time in high school, I found that the stress hormones caused me the most, stress, I guess, because there's a lot to remember here. Now you got the two responses to stress. The first one is the short-term response, and the other one is the more prolonged response to stress. The short-term response to stress is driven by the sympathetic nervous system. And ultimately, your sympathetic nerves target the adrenal medulla to release epinephrine. And epinephrine is one of those structures that, this is a word I like. I'm putting this word in, but it's not part of the exam, but I like this word. If you're going into the medical field, this is a good word. It's gluconeogenesis. Okay, don't have to know the word, but gluconeogenesis is the production of glucose from non-carbohydrate substrates. And that's what epinephrine does. Epinephrine makes sure that there's lots of glucose available for energy in that short-term response to stress. But even in the long-term response to stress, we have a more prolonged pathway. It's a hormonal pathway. The anterior pituitary releases a tropic hormone in response to a releasing hormone from the hypothalamus. The tropic hormone from the anterior pituitary targets the adrenal cortex. And the adrenal cortex responds by releasing hormones, glucocorticoids, mineralocorticoids. The most common glucocorticoid is cortisol. And cortisol, like epinephrine, engages in gluconeogenesis. It makes more glucose available in the blood. It does it by a variety of things, and non-carbohydrate substrates would be like things like amino acids. So making more amino acids available in the circulatory system enables cortisol to start converting those amino acids into glucose. This diagram, or something similar to this diagram anyways, from your textbook. It's the antagonistic hormones of insulin and glucagon. Insulin basically converts, makes cells permeable to glucose. So insulin is released after you have a meal, making cells permeable to glucose. If you can't fit any more glucose into the cells, then insulin converts glucose to glycogen, which is stored in the liver and some of the large muscles. And if there's glucose left over, then insulin will convert the rest of the glucose into adipose tissue. So hyperglycemia is a condition that usually affects the pancreas that isn't producing enough insulin. And so there isn't a way we can metabolize this glucose. We can't put the glucose into cells. We can't convert it into glycogen. Can't metabolize it into fat. So we get a condition called hyperglycemia. So hyperglycemia is a condition affecting the pancreas. It's caused by a reduction in the availability of insulin. So we get an increase of glucose in the blood, which means we get an increase of glucose in the urine. And since the glucose is not getting into the cells, it causes fatigue. So this diagram from your book is a great diagram because essentially everything on here you are expected to label. But one of the things that there's two types of questions that do come up quite often, and that is the composition of semen. Don't forget the testes is responsible for producing the sperm cells, which constitute part of semen, and also the exocrine glands, the seminal vesicles, the prostate glands, and the Cowper's gland. So these three exocrine glands, as well as the testes, they produce substances that make up semen. Now, the female reproductive structures, the important thing to do is to, of course, remember about the pathway the eggs need to take in order to get to the in order to get to the uterus. They're produced by the ovaries. It's very important as well to realize part of this course requires knowledge of comparative anatomy. So in other words, the ovaries are compared to the testes in males. The oviducts will be compared to the vas deferens. In males, the uterus doesn't really have the male version of it. That's about it, really. Yep, comparative anatomy, very important. This diagram here reminds you that ovulation occurs, with the ovum comes out of the ovary, and that fertilization occurs in the proximal end of the fallopian tube. A lot of people seem to think fertilization occurs in the uterus. It's important to realize that it has to occur really early because of the 10 days it takes to traverse the fallopian tube. So by the time it gets to the uterus, it'll be this multi-cell blastocyst that is mature enough to properly implant into the endometrium. And I'll give you this as a bit of a heads up as well. I mean, I haven't seen any questions on childbirth or sexually transmitted infections in this exam, so I'll just give you a little bit of a heads up on that. Probably shouldn't have done that. You should study it anyway, because maybe we'll just change the final exam on you just like that. All right, chapter 16 then is on cell division. Sometimes questions can be as easy as knowing the correct order of PMAT, prophase, metaphase, anaphase, telophase, and just the general processes that are going in to these stages. Prophase is where you see the chromatin becoming chromatids. Metaphase is where they line up along the equatorial plate. Anaphase is where the chromatids separate. And telophase is the stage just prior to cytokinesis, where we see each clump of DNA surrounded by a new nucleus. Meiosis is, you're going to be really, really comfortable with the steps of mitosis before you even start with meiosis. And meiosis is actually, it has two very, very important components within meiosis, important components for variation. The first one is the crossing over part of it. When crossing over occurs during prophase 1, this is where the non-sister chromatids exchange DNA, so to give us a very unique combination of alleles compared to what we had before. And the other thing, of course, is the independent assortment of chromosomes, the metaphase. The paternal chromosomes can be above or below, and there's, when you're talking about 23 pairs of homologous chromosomes, you got over 8 million different combinations. So between independent assortment and crossing over, we have basically an unlimited genotypic diversity. And of course, the big take-home message from meiosis is that we get haploid products. Our cells at the end are all haploid, and that'll lead us to the third mechanism of variation, which is fertilization. Having haploid cells means you've only got half the amount of DNA. We only have cells that are no longer containing genes that are no longer in homologous pairs. And so they need to be combined, fertilized with a cell that has the homologous partners. Non-disjunction is, again, this is something that you should be able to comfortably articulate. Non-disjunction leads to an odd number of chromosomes in the haploid cells. It's important to realize that non-disjunction does not cause Down syndrome, not on its own. I mean, you can indeed have a sex cell that at the end of meiosis is left with a homologous pair of chromosomes. But that's not Down syndrome. It only becomes Down syndrome if it's fertilized with a normal cell. That creates trisomy 21. Non-disjunction occurring in the first meiotic division really ruins all the cells. We end up having 2 cells that are n+1 and 2 cells that are n-1. But if non-disjunction occurs in the second meiotic division, 50% of the cells are expected to be normal. Also, interpreting karyotypes is important. This particular karyotype shows the normal arrangement of chromosomes for a female. Female because this, let me change my color. Changing it to white. Female because we have 2 X chromosomes. So this is a homologous pair of X chromosomes, therefore this is female. And everything else is paired up nicely as well. Well, this is normal. It's not likely you're going to get a question like that in an exam. If you're going to get a karyotype, be aware that it might have either missing chromosomes or extra chromosomes. And if this was male, of course, you'd have a Y, and then you'd have a little stubby little Y chromosome, but you'd have only 1 X chromosome. And you remember alternation of generations. It's the life cycle that you're most likely to get asked questions on. This particular diagram probably came from your book, but it's not likely you're going to get this diagram in an exam. So become as familiar as possible with the different ways that these alternation of generation diagrams can take, because basically they're all showing the same thing. You have a sporophyte generation or a diploid generation, and then you have a haploid generation. And typically you're asked which part are you supposed to know, which part produces cells by meiosis, which part produces cells by mitosis, and be able to identify them. Which part of the diagram do we see genetic variation, for example? Like, for example, I would see genetic variation occurring in the production of the sex cells, here. But I would not see genetic variation in the production of these sex cells here, because these are products of mitosis. Whereas in the diploid part, they are products of meiosis, and that's where we see variation. Then into Mendelian genetics. Now, I don't know if there are any monohybrid cross questions or not. I can't remember, but basically we started off talking a lot about monohybrid crosses, but in the end it's just far too simple. You have to build on that knowledge to get to talking about dihybrid crosses and X-linked heredity and epistatic alleles, which is a type of dihybrid cross. Epistatic alleles are the really alleles found on chromosomes that can control the expression of other genes. So in this particular question, for example, epistatic alleles control the expression of other genes. Gene B controls coat color in golden retriever dogs. Gene W at the other gene, a different chromosome, controls the expression of gene B. Now, it's always a good idea to kind of this, and when you're looking at a big blob of text like that, try to scrap of paper and just write out little notes to the side here. Write out, gene B controls coat color, gene W controls gene B. So, I mean, you can play this back later, but let me just all right, let's look at this particular question then. If W is homozygous recessive, B produces a black coat, little b produces brown. Any other instance, the dog is yellow. All right. What's the probability of black dog? So basically they're saying if we've got big W, then the dog is yellow. So it doesn't matter what the B situation is. If you see the big W, the dog's yellow. Only if we see the little w in the homozygous do we see color. So it's either going to be black or brown. This is the black allele. This is the brown allele. All right, so what's the probability of a black dog offspring from a cross between a double heterozygous yellow female and a brown male? So a double heterozygous yellow female is going to be this. I didn't need to be told this was a yellow female. The presence of a big W tells me this is a yellow female. And a brown male, well, that means it's got color. And the homozygous recessive B is brown. So this is a 4-square Punnett square. When you create your Punnett square, I have to draw slowly because it seems to get confused if I draw these lines quickly. All right, so little b, little w are the only combinations of alleles in the sex cells from the brown dog, but we got 4 different sex cells in the yellow. We got big W, big B, it's Warner Brothers. We got big W, little b, little w, big B, and little w, little b. So when you fill in the big W, big B, big W, this is going to be yellow because it's got a big W allele. This is also going to be yellow because it also has a big W allele. But this one is black. And this one is brown. So the question is saying, what's the probability of getting a black dog from this? That is 1 chance in 4, 0.25, or 1 chance in 4. Make sure that you're answering the question in, the way that, if it's a written response question, make sure that you're answering it the way it wants you to answer it. X-linked heredity. Basically, if it's a sex-linked trait, I should say, according to the title here, X-linked heredity are traits controlled by genes on the X chromosome. If it's X-linked, we only deal with X-linked traits in this course. Now, if it's recessive X-linked, we're going to see males with this condition. If it's dominant X-linked, then you're going to see more females. So keep that in mind if you're looking to interpret pedigrees. For example, when you look at this pedigree here, would you say this is indicative of X-linked recessive or X-linked dominant? Well, other than one male, it certainly looks like there are a lot more females with this condition. This would be an X-linked dominant heredity. Now, you've probably think, oh, hang on, this looked kind of familiar. Yeah, this was in your unit exam, for your unit 5 exam, I think. But it just really shows that you need to be able to look at a pedigree like this and be able to identify genotypes from these pedigrees. So for example, if you have these, here we go. We have these two individuals here. So individual 311 and 312. What is the probability of them having an offspring that is going to have the condition, for example? Well, we can see that individual 311 doesn't have the condition. So that means, and don't forget, it is a recessive trait if it's normal. So that would be his genotype. But the female has the condition. So she's XX. She has the condition. So she definitely has at least one dominant allele that is carrying the condition. However, is she heterozygous or is she homozygous? Well, you have to look at the parents. She would have, if individual 312, inherited her faulty X from her dad. Her mom was not carrying a faulty X. Her mom is homozygous recessive. So that means the offspring is homozygous recessive. And also, you can look at their offspring already. You can see the female here is carrying one of the X's from dad, but the other X is from mom. And since individual 49 is not affected, it means mom must have a good X to pass down. I did notice as well, somebody using the word carry, individuals who are carriers. Individuals are carriers are heterozygous. So carriers are heterozygous. Individuals who are homozygous for a condition, they're not carriers. They just simply have the condition and are homozygous for it. Blood typing is going to come up in this exam as well, not in the way that blood typing came up in Biology 20, but in Bio 30, we use pedigrees a lot for blood types, and it's a way that you can actually identify genotypes and infer genotypes of offspring. For example, this individual here, we know the genotype. Blood type O is little I, little I. But what's the genotype of this one? It could either be big IA. Big IA could be homozygous A, or it could be big IA, little I. So there's two possible genotypes. Which one is it? Well, this individual here is no help at all. It is obviously inheriting the A allele from the father. This individual here, though, tells me everything. This individual is also little I, meaning the little I comes from one parent, and the little I must also come from the other parent as well. So being able to pull genotypes out of pedigrees is extremely important. When you do some review, look for those kind of questions. Chromosome mapping is also important too. Definitely going to get chromosome map. People like chromosome mapping questions. Just to review with chromosome mapping, the line that you draw represents the outer boundaries of the two genes that are the furthest distance apart on the chart here. So between genes 2 and 3 is 24.3 distance. That means all the other genes, 1 and 4 rather, are going to sit between genes 2 and 3. So between 2 and 4 is 8.2. And between genes 3 and 1 is 13. So that's about here. So there we go. So in this particular case, what's the distance between genes 1 and 4? Well, you have 8.2 and 13. So that makes 21.2. This distance here is 24.3, the whole distance. So 21.2, 4.3, this makes 3.1. Anyway, if I'm going too fast, then just rewind and check my math on that. Okay, then we got into the biochemistry of biology, and I think in addition to the stuff on action potentials, I think the central dogma of biology, the transcription translation stuff, does tend to cause students a lot of problems because there's a lot to visualize here, and this type of biochemistry is very, is quite monstrous for a lot of people. So basically, with DNA, this nucleotide is really two nucleotides connected together at their nitrogen bases. And there's 4 different nitrogen bases, and they connect together using Chargaff's rule. For every adenine, there's a thymine. So the adenine and the thymines are going to be together, and the guanines and the cytosines are together. So for example, if we've got a piece of DNA and 35% of the DNA is guanine, what percent is adenine? These are very common questions because a lot of people either can do these really easily or really have a problem with these. If you have 35% guanine, that means you also have 35% cytosine, which is 70% altogether. Now, out of 100%, that means 30% must be made of equal amounts of adenine and thymine, which means we have 15% thymine and 15% adenine. Make sure to be comfortable with the differences between DNA and RNA. Most important, of course, is the fact that DNA has thymine, whereas RNA doesn't have thymine, it has uracil. And both thymine and uracil complement adenine. So be careful. Don't get tricked into trying to pair an adenine on a DNA molecule and being worried that you can't find thymine on RNA. And this is also very important as well. You're definitely going to need that part of your data book that enables you to translate mRNA codons into amino acids. So you can always assume that the DNA strand that they give you is the template strand from which messenger RNA transcribes. So if you're given a piece of DNA, don't do a complement of DNA first, unless the question specifically tells you to do that, and usually they don't. So get the DNA strand they give you, and then convert that to the messenger RNA transcript. It's the messenger RNA transcript that you read from your data sheet. And that translates into the amino acid sequence. So transcription occurs in the nucleus, translation occurs in the cytoplasm. Be comfortable with the order these things occur. These would make great numeric response questions. They'll give you a whole bunch of steps, and then you got to put them in the correct order. So be comfortable about the order that these things go in. Mitochondrial DNA inheritance patterns. Basically, the mitochondrial DNA in everybody was inherited from your mothers. And the reason is because during fertilization, it's the mother's egg cell that is carrying the mitochondria and therefore carrying the mitochondrial DNA. It gets passed down to the offspring. The only thing that dad gives up during fertilization is the nucleus, and there's no mitochondrial DNA in the nucleus. Also, the biochemistry of recombination technology is going to come up as well. There are DNA recombination enables things like transgenic technology, for example, where we can incorporate a gene from one organism into the genome of another organism, of a different organism altogether. There's more information on this particular diagram than you need to know, but basically we're using restriction enzymes to cut out a piece of DNA that contains the required gene and then paste it into a genome of a bacteria. The bacteria has been prepared by being cut with the same restriction enzyme, and then we use ligase enzymes to glue the DNA together. So the key terms, which I don't see written on here, restriction enzymes, and ligase. So restriction enzymes are the molecular scissors for DNA, and ligase is the molecular glue, glues them back together. One of the technologies that you were hopefully read about is the gel electrophoresis, and that's exposing DNA to multiple restriction endonucleases to really shred the DNA and then put it into gel electrophoresis. And the different strands will separate along the length of the gel. So it's going from one end to the other. And the larger ones have a much harder time navigating their way through the gel than the smaller ones. So the smaller fragments will move through the gel much more quickly than the larger ones. There's going to be Hardy-Weinberg calculations. There's only really one way to do these questions, and so really once you master that one way, you could make very short work of this. You need to have both of these equations. You're given this one, but you're not given this one. So when you come across this question, write this down to remind you. Now, you're almost always going to be given this. Oops. You're going to be almost always given this one, Q squared. It's either going to be a percentage, in which case you divide it by 100 to get the frequency, or you could be given raw numbers, in which case you divide it by the total number in the population to get the frequency. Either way, you're going to have to square root Q squared to get Q. And once you've got Q, 1 minus Q is going to give you P. And then once you have P, you can solve either of these. Square it, you get P squared. Multiply it by 2, multiply it by Q again, and you get 2PQ. Don't forget the heterozygous genotype is going to be 2PQ. Homozygous dominant is P squared. Homozygous recessive is Q squared. Now, with these types of questions, they typically accompany a bit of a blurb that you have to read about and then describe what the ecological relationships are between the organisms that were described. So be comfortable at the difference. Predator-prey relationships. Now, I put plus minus here because the predator benefits while the prey doesn't. But the parasitism is the one at the bottom there. It looks like it has the same kind of thing. Is the parasite the predator and the prey is the host? And no, not really. There are very complex relationships that occur between organisms. So, the parasite. Think of the parasite as being something which only works to take away the nutrients and benefits from the host. The predator-prey is a bit more complex and involves larger organisms. The predator species can actually suffer if there's not enough prey, whereas with parasitism, the host species don't suffer if there's no parasites. Mutualism is where both organisms benefit, and commensalism is where you see one benefit and the other one neither benefits nor suffers. Again, practice looking at those questions. You're not going to get a question that's going to require you to find anything like this. It's going to give you a scenario, with different animals, and then have you explain, well, what's the relationships going on here? And finally, the R versus K selected species. Now, the R and the K, they don't stand for anything. They're just constants used in mathematical expressions, where R represents maximal growth rate and K represents carrying capacities. Organisms that are specialized in breeding at carrying capacities are those organisms that like environments that are stable. They exist in numerical harmony with their environment. They're typically bigger organisms. They don't breed profusely. They have very low biotic potentials. Whereas the R-selected species, they live in very unstable environments. They have very high biotic potentials. They produce a lot of offsprings. Anyway, this table here separates the two, but generally have an idea between them. And it's sometimes very difficult, but you're going to realize that there are some organisms that can actually show both types of selection. For example, there are some fish, for example, that will live to very old age. Well, that sounds like a K-selected species, and yet these fish will produce an awful lot of offspring during spawning. So that's an R-selected. So. All right. Well, that's it, folks. That was an hour. If you have any concerns or questions, I'm going to be around actually for until August 23rd, actually, and then Phil comes back. But if you have any concerns or questions, anything you want explaining, any help you need, please drop me a line. And after the 23rd, drop Phil a line, and we'll be happy to answer any questions you have before you take that final exam. Okay, enjoy your studies. Have a great rest of your day, folks.